Hello guys, welcome to Lab 1 hand washing experiment. I'm Sofia Guerra and my lab partner is Mayra Landeros. And we're going to be explaining our hand washing experiment techniques and results. First, for introduction. Purpose of this experiment was to investigate whether washing hands with soap is more effective than washing with only water in reducing microbial presence. Hand washing is a simple but powerful public health tool that directly impacts infection control, which is especially important in healthcare and everyday hygiene. This connects to broader microbiology and health science concepts because it demonstrates how microbes interact with our skin and how different cleaning agents, soap versus water in this case, impact bacterial removal. We expected to see differences in bacterial colonies between the water-washed plates R1 and R4, R1 through R4, and the soap-washed plates L1 through L4. Specifically, soap was predicted to be more effective due to its chemical ability to break down oils and membranes, while water primarily relies on mechanical friction. This experiment also allowed us to consider real-world applications, such as how the results might differ if hand sanitizer were used instead of soap and water, or how friction rubbing plays an essential role in dislodging microbes from the skin. For our materials, we had four blood agar plates labeled R1 through R4 for right thumb and L1 through L4 for the left thumb. We used sterile cotton swabs, saline solution, soap and sponge, deionized water, and a staining tray. For our methods, our procedure was that we started off with the right thumb, which was washed with water only. R1 was inoculated before washing, R2 after 1 minute of washing, and R3 after three total minutes, and R4 after 6 total minutes. The left thumb was washed with soap and water. L1 was inoculated before washing, L2 after 1 minute of scrubbing with soap, L3 after 3 total minutes, and L4 after 6 total minutes. Each thumb was rubbed or scrubbed under the assigned conditions, rinsed, and swabbed. The swabs were inoculated onto blood agar plates, which were incubated for bacterial growth. Method continued. This method allowed us to directly compare the rate of bacterial reduction between water alone and soap, as well as the importance of washing duration. It also highlights limitations of the setup, such as possible contamination, uneven scrubbing, or accidental mixing of soap runoff with the water only side.
Now for results and observations. As you can see for the left thumb, before washing showed higher bacterial growth. As for the second time after 1 minute, showed noticeably fewer colonies, confirming that soap was already effective after short exposure. As for the total after 3 minutes and total after 6 minutes, some too little bacteria were visible. This suggests strong soap effectiveness through experimental error may have contributed possible soap runoff or inconsistent swabbing. For the right thumb, before washing, it showed very minimal bacterial growth. After the 1 minute, colonies were reduced but were less present than on the left thumb the second time. For the 3 minutes and 6 minutes, very few colonies were visible, suggesting that extended friction and rinsing can still significantly reduce microbes, but perhaps not as effectively or consistently as soap. Based on the graph here, soap reduced bacteria more quickly. Both methods showed minimal growth by 6 minutes. Unexpectedly, low bacteria in later plates may reflect procedural error. Unexpected results by the final stages R3, R4, L3, L4, both thumbs showed little to no bacteria. While this might reflect effective washing, errors such as not allowing swabs to dry fully or uneven swabbing could explain the near absence of colonies. Interpretation. Soap demonstrated a clear advantage in the early stages of washing. Late water alone was less effective initially but could still reduce bacteria over time. Friction from scrubbing appears to play a critical role in both methods since mechanical removal helps dislodge microbes. Conclusion. This experiment showed that soap is more effective than water alone in reducing bacterial presence, particularly within the first minute of washing. The left thumb with soap consistently showed a sharper decline in colonies compared to the right thumb with just water. By 6 minutes, both soap and water produced plates with very little bacterial growth. Through possible procedural errors may have influenced these results. The findings suggest that soap provides chemical benefits beyond the mechanical action of friction, making it more reliable for hand hygiene. If hand sanitizer had been tested instead, results may have shown rapid bacterial reduction as well. Through sanitizer effectiveness depends on alcohol concentration and thorough application. From a public health perspective, this experiment reinforces recommendations for using soap and water for hand washing to prevent the spread of infectious diseases. It also demonstrates how simple lab experiments can simulate real-world hygiene practices. Though improvements such as controlling for soap runoff, standardizing swabbing technique, and repeating trials would strengthen the results. Overall, this experiment connected microbiology principles with practical hygiene behaviors. It highlighted the significance of friction, the superiority of soap, and the importance of consistent hand washing as a foundation of infection prevention. Thank you.